Good morning, students. Today we are discussed about plant hormone ethylene. Earlier we discussed about auxin and zebranin hormone ethylene. It is the simplest olefin. Today we start with structure, biosynthesis and measurement of ethylene. Ethylene is the simplest olefin and its molecular weight is 28. It is lighter than air under physiological conditions and readily undergoes oxidation. Ethylene can be produced by almost all parts of higher plants, although the rate of production depends on the type of tissue and the stage of development. It is usually measured by gas chromatography. Ethylene production increases during leaf abscission and flower senescence, as well as during fruit ripening. Any type of wounding can induce ethylene biosynthesis, as can physiological stress such as flooding, disease, temperature, or drought stress. In addition, infection by various pathogens can also elevate ethylene biosynthesis. The amino acid methionine is the precursor of ethylene and 1-aminocyclopropane, 1-carboxylic acid, serves as an intermediate in the conversion of methionine to ethylene. We will see, the complete pathway is a cycle taking its place among the many metabolic cycles that operate in plants. Ethylene biosynthesis is promoted by several factors. Ethylene biosynthesis is stimulated by several factors including developmental state, environmental conditions, other plant hormones, and physical and chemical injury. Ethylene biosynthesis is also varies in a circadian manner, peaking during the day and reaching a minimum at night. First is fruit ripening. As fruit matures, the rate of aminocyclopropane 1-carboxylic acid, or we can say ACC, and ethylene biosynthesis increase. Enzyme activities for both ACC oxidase and ACC synthesis increase, as do the messenger RNA levels for subsets of the genes encoding each enzyme. However, application of ACC to unripe fruits only slightly enhance ethylene production, indicating that an increasing in the activity of ACC oxidase is the rate limiting step in ripening. Next is stress induced ethylene production. Ethylene biosynthesis is increased by stress conditions such as drought, flooding, chilling, exposure to ozone, and mechanical wounding. In all these cases, ethylene is produced by the usual biosynthetic pathway, and the increased ethylene production has been shown to result, at least in part, from an increase in transcription of ACC synthase messenger RNA. This stress ethylene is involved in the onset of stress response such as abscission, senescence, wound healing, and increased disease resistance. Next is circadian regulation of ethylene production. The circadian clock regulates the biosynthesis of ethylene in a number of plant species. There is generally a peak of ethylene evolution at midday with the through in the middle of the night. This regulation likely results from the transcriptional control of a subset of ACC synthase genes which is mediated by TOC1 and CCA1 clock in Arabidopsis. Next is auxin-induced ethylene production. In some instances, auxin and ethylene can cause similar plant responses, such as induction of flowering in pineapple and inhibition of stem elongation. These responses might be due to ability of auxins to promote ethylene synthesis by enhancing ACC synthase activity. These observations suggest that some responses previously attributed to auxin are in fact mediated by the ethylene produced in response to auxin. Following application of exogenous in IAA, the transcription of multiple ACC synthase genes is elevated and increased ethylene production is observed. Inhibitors of protein synthesis block IAA induced ethylene synthesis, indicating that the synthesis of new ACC synthase protein caused by auxins bring about the marked increase in ethylene production. Various inhibitors can block ethylene synthesis. Inhibitors of hormone synthesis or action are valuable for the study of biosynthetic pathways and physiological roles of hormones. The use of inhibitors is particularly helpful when it is difficult to distinguish between different hormones that have identical effects in plant tissues, or when a hormone affects the synthesis or the action of another hormone. For example, ethylene mimics high concentration of auxins by inhibiting stem growth and causing apinesty, downward curvature of leaves. Use of specific inhibitors of ethylene biosynthesis and action made it possible to discriminate between the actions of auxins and ethylene. Studies using inhibitors showed that ethylene is the primary effector of apinesty and that auxin acts indirectly by causing a substantial increase in ethylene production. Inhibitors of ethylene synthesis, amino ethoxyvinyl glycine AVG and amino oxyacetic acid AOA block the conversion of to ACC. AVG and AO are known to inhibit enzymes that use cofactor pyridoxal phosphate, including ACC synthase, alpha amino isobutyric acid, and cobalt ions also inhibit the ethylene biosynthetic pathway, blocking the conversion of ACC to ethylene by ACC oxidase, the last step in ethylene biosynthesis. Inhibitors of ethylene actions. Most of the effects of ethylene can be antagonized by specific ethylene inhibitors. Silver ions applied as silver nitrate or as silver thiosulfate are potent inhibitors of ethylene action. Silver is very specific. The inhibition it causes cannot be induced by any other metal ion. Carbon dioxide at high concentrations also inhibits many effects of ethylene, such as the induction of fruit ripening, although CO2 is less efficient than silver ions. This effect of CO2 has often been exploited in the storage of fruits whose ripening is delayed at elevated CO2 concentrations. The high concentration of CO2 required for inhibition make it unlikely that CO2 acts as an ethylene antagonist under natural conditions. The volatile compounds trans-cyclooctane but not its isomer cis-cyclooctane is a strong competitive inhibitor of ethylene binding. Trans-cyclooctane is thought to act by competing with ethylene for binding to receptor. 1-methyl cyclopropane (MCP) binds almost irreversibly to the ethylene receptor and effectively blocks multiple ethylene responses. This nearly odorless compound has been marketed under the trade name ethyl block and is used to increase the shelf life of cut flowers and of some fruits such as apples. So it's finished. Thank you, students.